Cure, Connect, Transform. There is no greater embodiment of Mayo Clinic's plans to revolutionize healthcare than the Center for Individualized Medicine. We discover, translate, and apply new individualized medicine advances in patient care. Using genomics and multiomics could lead to vast advancements in knowledge of the causes and risk factors for many diseases. Our researchers and clinicians work together to accelerate discoveries in 4 strategic areas. Simplify and scale. In Genomics in Action, we work to transform the practice by providing the knowledge, tests, and tools needed to make it easier for clinicians to integrate genomics into routine patient care. Individualizing cures with genes, bugs, and drugs. Cutting-edge therapies being developed with high-definition therapeutics are driving precision medicine science beyond diagnostics to provide personalized cures. Connecting data to practice. Our exciting work with Omics Data Platform is centralizing genomic and multiomic data into a single repository, which will allow for the most individualized medical approaches possible. A multi-omics future. In Beyond DNA, we are advancing patient care by using molecular information from the microbiome, epigenome, exposome, and more.
The Center for Individualized Medicine and Mayo Clinic are in a unique position to fuel new discoveries to improve care for patients powered by sophisticated research and artificial intelligence. From providing the best individualized care to addressing the world's most challenging healthcare problems, Mayo researchers here are relentlessly pursuing discoveries that deliver hope and better health to people today and for generations to come. Thank you.
Good afternoon, everyone. My name is Margot Cousin, and I'm a PhD research associate at the Center for Individualized Medicine at Mayo Clinic in the Translational Omics program. I would like to welcome you all to the Center for Individualized Medicine Grand Rounds. The CIM Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation and demonstrate how individualized medicine is being translated into practice to meet current and future patient needs. Today, we are highlighting the theme of high-definition therapeutics, which looks to drive precision medicine science beyond diagnostics to provide personalized cures. My current research focus on individualized therapies for rare disease patients was in no small part inspired by our speaker's groundbreaking work. So it is my pleasure to introduce today's presenter, Dr. Timothy Yu. Dr. Yu is a neurogeneticist at Boston Children's Hospital and Harvard Medical School. He obtained his undergraduate degree in biochemistry at Harvard College, which inspired an early preoccupation with structural biology and rational drug design. Those interests shifted during his MD PhD training at UCSF, in which he focused on neuroscience and C. elegans genetics. After completing neurology residency at Massachusetts General Hospital and Brigham and Women's Hospital, he joined the faculty at Boston Children's Hospital, where he has found his work to have come full circle, combining human genetics, neuroscience, and molecular medicine. He currently serves as a staff physician and researcher in the Division of Genetics and Genomics and the Department of Neurology at BCH, associate professor at Harvard Medical School, and associate member of the Broad Institute. Today, Dr. Yu will be speaking on piloting pathways for interventional genomics in orphan neurologic disease. The genomic revolution has shortened the diagnostic odyssey for rare diseases, but treatments remain scarce. So to meet this need, novel platform technologies allow us to develop drugs that are tailored to specific mutations. But these mutations may be present in only as few as a single individual. So what do you do when the best available therapeutic strategy is an experimental drug for one? How do you responsibly gauge the risks and benefits for these first-in-human interventions? These technologies pose both opportunities and challenges and begin to blur traditional distinctions between what we consider a drug versus, say, a procedural intervention. Dr. Yu will discuss these questions through the lens of his experience developing tailored drugs for a range of fatal orphan neurogenetic conditions. So throughout this talk, please use the Q&A in Zoom to submit your questions. We have planned Q&A at the end of the session for those who have not yet claimed continuing education credit. A message will be sent out in the chat shortly. So now please join me in welcoming Dr. Yu. Thank you.
Thank you so much for the very kind introduction, Margot. And it's a real pleasure to be here, if only virtually for the moment. and to get the chance to share our work and to talk with you about its implications and to really and to engage in a discussion with all of you. Let me go ahead and get my slides up and running. First, as Margot indicated, I am a neurologist and a geneticist at Boston Children's Hospital. I spend my time primarily doing research that involves patients very intimately in a way that I hope to illustrate during this talk. And as mentioned, this talk will be about piloting pathways for patient-customized oligonucleotide medicine. First, my disclosures. I disclose the following. I have served on advisory boards ad hoc for a few commercial companies shown in the top row, and have the pleasure of volunteering for a number of nonprofits, all working in the rare disease space. By way of background, our research group at Boston Children's has focused on these 4 domains shown on this slide. We really began our work thinking hard about how to use human genetics to interrogate basic mechanisms of human brain development. That also parlayed itself into a strong early interest in neurodevelopmental diseases such as autism, and using a large-scale sequencing to try to identify genes that underlie this very complex disorder. Along the way, we've also spent time in tool development, thinking hard about protocols for genome analysis and algorithms for genome interpretation. But it's this 4th category in the bottom right quadrant, which is the newest category and intense focus of our group, that I'm going to be focusing on today. And our work in this space was really motivated first by this conceptual challenge on this slide, that we've been very lucky over the last 15 years to live in a time when tools in genomics allow us to greatly accelerate the diagnosis of patients with rare disease. And we were very early adopters of this high throughput sequencing technology, regional capture, exome capture, then whole genome sequencing. It really fundamentally changed the way that we go about discovering genes and then diagnosing patients. And as we've lived through that wonderful revolution, it's increasingly focused our attention, though, on the remaining challenge, which is that going from a diagnosis to developing a treatment still takes a very long time. This, however, is beginning to change. And in this lesson was really one that we learned very fortuitously some 3.5 years ago, when we published the following report, Patient Customized Oligonucleotide Therapy for a Rare Genetic Disease. This is a paper that we've become known for, and I'd like to go over it in brief to describe the major findings and why it's motivated us so much in this space. This was an unusual case where we encountered a 6-year-old girl who was suffering from several years of insidious neurologic symptoms. These began with vision loss, progressed to some mild clumsiness, which turned into stumbling and then tripping and then falling. And then those symptoms all began around age 3.5 and then progressed slowly to age 4, age 5, and around age 6, she began suffering a much more severe neurologic progression with loss of words, loss of ADLs, and loss of the ability to walk. And she had been worked up by Dr. Austin Larson at Colorado Children's Hospital. And she had been conferred a very, very unfortunate diagnosis. A MRI showed cerebellar atrophy. Skin biopsy showed that she had abnormal fingerprint-like inclusions. And the nature of these inclusions indicated lysosomal buildup of a substance called steroid lipofuscinosis, which was pathognomonic for a condition called Batten disease. Batten disease is a family of disorders that come in 14 different types. Each of those types is associated with recessive mutations in key lysosomal genes. And unfortunately, this is a condition that leads to progressive neuronal cell death and ultimately is fatal for the patients affected. And this particular patient, Mila, had a subtype CLN7 that was a rare subtype of an already rare family of diseases. CLN7 Batten disease itself has been reported in some 70 patients in the literature. And unfortunately, in the classic form, which is consistent with what her clinical presentation was showing, this predicted death by age 11. Now, the wrinkle in this case, however, that led to our engagement was that she had received this clinical diagnosis, but at the molecular level, she only had half of a diagnosis. Clinical genetic testing arranged by Dr. Larson had shown that she had a heterozygous mutation in the CLN7 or MFSD8 gene, but the second mutation was missing. And that actually turned out to be quite fortuitous for her. In an attempt to try and clinch the diagnosis, the family reached out, again, this is in 2017, for help on the internet. Their team had correctly told them that to find the missing second mutation would likely require something called whole genome sequencing. And back in 2017, that was hard to come by. So it came to be that we connected, of all things, via social media through a group called the Physician Mothers Group to this message that led us to this family. My wife actually saw this on a Friday evening and described this case of a family with this proposed diagnosis that they needed assistance to confirm. And at that time, we were working very hard on using whole genome sequencing, which was still quite in its early days. And we were developing algorithms for how to find difficult to discover mutations using whole genome sequencing. And this clinical need lined up very well with the research that we were going, that was ongoing in our lab, and we offered to help. What we reported to the family and in our paper is that whole genome sequencing revealed an unusual finding. We found via whole genome sequencing that there was a cluster of unusual deep intronic reads within her CLN7 gene that were far away from exons and therefore missed by traditional gene sequencing. And this cluster of mismapped reads in intron 7, shown here circled in yellow in the top track in our patient, appeared to have been inherited from her mother. And that was significant because the known clinically significant heterozygous mutation that had been previously found in her was inherited from her father. So this unusual set of reads looks like it could be possibly part of the missing piece. And when we looked more closely at this cluster of reads, we saw that there were unusual features to these reads. First of all, there were sharp breakpoints in these reads that were demarcated with a 14 nucleotide gap. Those breakpoints are showed in the dotted vertical lines here. Those breakpoints, it turns out, are caused not by actual breakpoints of the reads themselves, the DNA that was actually sequenced, but these were areas where the sequence diverged so much from the human genome reference that our mapper didn't know what to do with it, so it just clipped it off. When we turned that feature off, we discovered that these reads were actually chimeric reads, where after these dotted line breakpoints, the sequence diverged into foreign DNA sequence, which is not normally found in this MFSD8 or CLN7 genomic locus. And on one side, there were poly-T sequences. On the other side, there were hexameric repeats, GGG, AGA, GGG, AGA, over and over and over again. Through some relatively esoteric literature searching and consulting with colleagues like Alice Lee, we deduced what this actually was telling us, that this unusual configuration of reads was indicating to us that there had been a DNA insertion at this site, deep within this intron, and an insertion that on one side began with the poly-T stretch, and on the other side ended with the hexameric repeat stretch. And if you do some DNA origami and draw things out in a fashion that I don't have time to do, it would actually create this pattern shown here. Again, the details truly aren't that important, but it's simpler to visualize this as a large stretch of 2,000 nucleotides, which had inserted themselves into intron 6 of the CLN7 gene. And it turned out that this insertion had been the result of hopping of a piece of mobile DNA, something called an SVA retrotransposon. And these are genomic viruses that can hop around in our genomes, and they can be a cause of monogenic disease. They are relatively underappreciated, except to a few dedicated academic genetic cognoscenti, but they are part of the missing dark matter of clinical conditions where sometimes we know we've clinched the diagnosis, but we can't find the mutations. And this seemed to be the case here. How is this actual DNA insertion breaking the gene? How is it actually affecting the impact of the function? Well, it turned out that this insertion was creating a new splice site and disrupting proper gene assembly. And that was a hypothesis that we first came up with and then confirmed using appropriate RT-PCR and then RNA sequencing of the transcriptome of our patient. It also, however, led us to a therapeutic hypothesis. And that therapeutic hypothesis was basically us asking ourselves the question, here's a situation where the coding region of our gene is entirely intact, but there has been insertion of a foreign DNA element here that creates a novel exon. What happens if we could silence this abnormal exon? What would happen if we could silence this abnormal splice event that created this abnormal exon with something called an antisense oligonucleotide? Could we silence the splice event and restore the normal processing of this gene and restore gene function for our patient? And so that's what we set out to do. Our inspiration for this was a technology called antisense oligonucleotides, which had just proven themselves in the clinic in 2017 for a disease called spinal muscular atrophy. Many of you, I'm sure, are well aware of it. This was remains today one of the most remarkable clinical success stories, clinical and scientific success stories that I can think of, where administration of a 18 nucleotide synthetic antisense oligonucleotide, the synthetic stretch of RNA, given by spinal tap into infants with this devastating neurodegenerative condition, was shown to be able to restore the proper splicing of the SMN2 gene, to alter the splicing patterns, restore normal functional protein, and rescue the clinical phenotype of patients, infants with this condition. And this drug remains the inspiration for many folks. In our case, it was the inspiration for saying, can we actually do this exact same thing for our patient? We did do that. That's why I showed you the publication at the outset of the talk. And let me just show you the pillars of the experimental plan upon which that effort was accomplished. First, using the Spinraza template, we were able to do a bioinformatic analysis of the splice regions, the splice enhancing and modulating regions around our patient's mutation. We were able to identify sites that promoted missplicing, and we were able to then design patient-customized oligonucleotides to cover those splice sites and to block them and to inhibit their function. And we were able to test our designs using patient cells that were donated from skin biopsies and from lymphoblast draws. And we could use our patient's own cells in this way as a test bed to interrogate her very unusual mutation, to treat them with antisense oligonucleotides, and to do appropriate RNA studies to look at both missplice variants and normal splice variants of this gene, and see if it could boost normal splicing at the expense of mutant splicing. And we were able to do this using techniques like RNA sequencing and transcriptome profiling to quantitatively assess the amount of normal splicing that was occurring in our patient, which was only between 7 and 11%, as shown in these top two rows here. And then we were able to use those same techniques to ask the same question after treating our patient cells with our patient-customized oligonucleotide, showing that we could boost the levels of normal splicing by up to 5 times. And finally, we were able to take this effort and extend it not just to correction of molecular biological RNA transcripts, but to improve cellular health. We collaborated with Joe Mazzuli's lab at Northwestern and showed that electron micrographs of our patient's cells showed the accumulation of these abnormal white bubbles representing abnormal storage material in the cytoplasm of her cells. And Jo was able to show with us the treatment of her cells with our oligonucleotide in just 24 hours led to a massive reduction in the number and size of those abnormal storage vacuoles, as well as improvement in a number of other biochemical assays that his lab pioneered. All of this really led us to the really interesting clinical question. With this sort of proof of concept scientific data in hand, would it be appropriate to actually propose to try to treat our patient? And this was a decision that we thought about quite hard, and we enlisted consultations with our ethics committee at the hospital and colleagues. And these were some of the factors that compelled us to actually move forward. First, reflecting on the fact that this was obviously a devastating, fatal, and orphan disease, it meant that the risks of treatment here had to be weighed against the risks of the disease itself, which from the natural history was assured to be fatal. Number two, importantly, the proposed treatment was really precise, and it rested on a precise diagnosis and a very specific and demonstrable impact, a functional impact that could be tested in our patient's own cells. And arguably, we felt, and others agreed, that this sort of genetically targeted and precise treatment arguably boosted the odds of positive impact. Certainly no guarantee, but this was not, for instance, a small molecule that we were hoping might just tip the scales of a complex pathway just enough to help a patient. This was a much more targeted and specific therapy. Finally, point three, from a public health standpoint, the FDA's charge is to protect the public in these sorts of investigational efforts to make sure that the drug supply is appropriately quality checked before exposure to patients. We were, in this case, working with a patient with a very unique mutation found only in her, and she was the only Batten patient we knew of with this condition. Turns out her mother and her grandmother were carriers, but there were no other Batten patients who had this specific mutation. And what that meant was the public health exposure to this. This was not a broadly marketed drug trial that we were planning to launch. It meant that with, again, appropriate and careful consent of the family, we would be asking for permission to work with this one particular family, much more under an individual consent, really much more almost like a surgery than, say, an enrollment in a broad trial per se. This effort was advanced in collaboration with the FDA, as well as a number of FDA, ex-FDA veterans who served as advisors for us. And with folks like Lauren Black and Sarah Goldkind and the folks here, we were, and folks at the FDA itself, they were actually demonstrated great flexibility with us in seeking to file this under an expanded access protocol. There were a number of challenges. This was the first time using this particular route for a brand new drug. Usually expanded access is about expanding the indications of an already approved drug to a new purpose. But this, to our knowledge, was the first time that a drug had been made explicitly for one patient on the basis of his or her genetic sequence. And so there were important challenges to be navigated. But I don't have time to go into the details. With tremendous help from many, we were able to come up with reasonable strategies to accommodate those. And our results from this trial, which we published in, as I described, really exceeded reasonable expectations. What we found was strong evidence of target engagement shown by objective measures such as seizures. Our patient, prior to initiating this investigational therapy, was having between 10 and 30 seizures per day. These were lasting typically about 2 minutes in length. And what we saw is that after enrollment in the trial, the number and the duration of her seizures shrunk remarkably. She started treatment in January 2018. By June of 2018, the number of seizures she was exhibiting had shrunken to 0 to 10. And the duration of the seizures had gone from on an average of 2 minutes down to on an average of just a few seconds. This chart shows those trends over that first year. In the second year, you can see that we decided to increase the dose a little bit, and we saw what we appears to be a further slight reduction in seizure frequency, although they never went away completely. Now, I wish I could say that this was a fully successful trial. The two objective outcome measures that we selected for this were, on one hand, seizures, because we could document those on daily records from diaries, but also through 24-hour video EEGs, which tracked and corroborated the previous slide's changes. The other objective measure that we followed was brain volume loss. And I am sad to report, though, that despite the seizure improvements, brain atrophy had been evident for 3 years of serial MRI images prior to enrollment in our trial. And while it's possible that that volume loss might have slowed, we can't say that with any certainty. And it certainly did not stop. And so this slow progression of brain volume loss ultimately was associated with a gradual clinical decline, such that in year 3 of our trial, even though we think that she was doing better than most patients at this temporal stage in her CLN7 disease, the family, with clinician support and our support, made the very difficult decision to transition to hospice because her level of responsiveness and interactiveness and quality of life had crossed a threshold that all felt was appropriate for her. Nonetheless, this work, which has really ignited a lot of renewed interest in this idea of individualized medicine, and to put a particular spin on that, a particular flavor of individualized medicine that people refer to as programmable medicines. The concept is that with drugs like Spinraza, also known as nusinersen, shown up on top, an 18 nucleotide sequence that can target the SMN2 gene and alter splicing. And then closely related drugs like Milasen, which were made using the exact same chemistry, the exact same experimental approach, the exact same manufacturing process, formulation, and delivery, but just a different sequence, plus it was slightly longer. Is this a trick that could be pulled over and over again? Can we use these medicines as programmable units to repurpose the platform and use them to make drugs for many, many conditions very, very quickly? We know that there are significant advantages to the ASOs that spurred us to try this in the first place, and those advantages are listed on the slide. ASOs, because they are targeted to their sequences by simple Watson-Crick base pairing, are rapidly customizable. The processes for manufacturing ASOs have been well mature for now some 40 years, and they're quite inexpensive to manufacture. It turns out that the delivery of antisense oligonucleotides to the brain and spinal cord is especially streamlined. Taking these ASOs and dissolving them in artificial CSF, which is basically a pH buffered salt solution, is sufficient to get widespread uptake throughout the brain without viruses or lipid nanoparticles or other adjuvants. And there are now several, over a dozen standard clinical trials using antisense oligonucleotides in various in-human studies. And so there's a growing safety and efficacy record for these molecules that will allow us to build on our experiences and leverage data across different trials. And so that's really spurred us to talk about this concept. Can ASOs be an example of a platform that is maturing? And could it be ready for use as a platform for individualized medicines, or if not even N of one, at least N of few or N of small? And could we use these to address this looming problem, which we're all aware of, with some now 7,000 different genetic diseases that are known? The great majority of these are actually too small for traditional commercial interest. Can these efforts like Milasen be used to approach them and to make them now actually navigatable. Can individualized efforts, even like Milasen, even if they are only for small numbers of individuals, can we learn lessons from those efforts that might validate a particular gene target? Can we learn lessons that might allow us to probe a critical therapeutic window? Our experience with Milasen arguably indicates that treating at that stage in Batten disease might be sufficient to improve quality of life in terms of seizures, but sadly may not be sufficiently early enough to arrest the overall neurodegenerative brain atrophy which may have already set in. And finally, again, the core platform concept is that if one can keep constant as much as possible the chemistry, the mechanism of action, the administration route, but change as little as possible, just the sequences, then can this be made in a way that's a scientific and aggregatable data set to share learnings across these different drugs and support this approach more broadly. We had meetings with the FDA to talk about this in the wake of launching our Milasen effort. It seemed to us that if we had done this, that there would likely be many others to follow. And one of the points that we discussed in that very productive meeting was the importance of trying to do this in a very rational, a very transparent, and very controlled fashion, so that we could establish if this actually could work. With an N of 1, it's difficult to establish efficacy, even though with the data that I showed. How repeatable is this experience? And truly, even if our drug appeared to be safe and well tolerated by our patient, we cannot guarantee that that would be true for every drug made from this chemical class. So what are these critical safety parameters across the platform? And then importantly also, this effort to make Milasen was really a Herculean research scientific effort, but is there a path to making this type of effort accessible, sustainable, less expensive, so that it can be scaled and given to more individuals if it can be shown to work? As we had suspected, since the publication of our effort in 2019, there have been a number of other now published examples that have used antisense oligonucleotides to accelerate interventional trials for individuals in a patient customized way. And these examples here followed in the years to follow an FUS ALS case, another C9orf72 ALS case. These were advanced by colleagues and close collaborators, Neil Schneider, John Watts, and Bob Brown at Columbia and UMass, respectively. And they represent, I think, the tip of an emerging iceberg of other individualized ASO efforts that we know are underway. We personally know of several dozen folks who are working in this space, several dozen projects to try to advance this type of technology. We ourselves have been working on a series of individualized follow-up ASO trials, and I'll tell you about 2 of these. Since our CLN7 Batten case, we worked on ataxia telangiectasia and neonatal epilepsy. And then there are many other cases that we're working up, at least at the cellular level, to see whether or not there might be something that could be suitable for a clinical application. Our ataxia telangiectasia case began with this young child who was, unfortunately for her, accidentally diagnosed with a fatal genetic disorder at birth. She had an abnormal newborn screen. She had a low T-cell receptor excision count, a newborn screen for severe combined immunodeficiency. It turned out that her low Treg count actually was not caused by SCID. It turns out that it was actually caused by a defect in the DNA repair pathway that was caused by mutations in the ATM gene. And ATM gene mutations cause a condition called ataxia telangiectasia, which unfortunately for these children leads to progressive cerebellar neurodegeneration. These children are wheelchair bound by age 10 and typically die by young adulthood. Now, this sad situation for this young infant was actually had possibly a silver lining, or at least a silver lining opportunity. One of her mutations, her ATM mutations, looked to us very much like the mutation that our Batten patient had. It created a novel splice site and had no impact on the function of the protein otherwise. And so we set out to do the same thing as we had done for Milasen. We conducted ASO design and then screening using patient cell lines to try to rescue her ATM splice defect. This slide shows the sequence of ATM exon 53. And in the red box is the splice donor site created by her specific mutation, a GC to GT change that creates a new GT splice donor site. And that use of that splice site leads to truncation of exon 53 and a mistranslated protein. We covered this region with antisense oligonucleotides, and we're able to find several that could restore normal ATM splicing in a dose-dependent fashion. Here are two bands on a gel, the lower of which is abnormal, the top of which is normal. And you can see a nice dose-dependent restoration of the normal splice pattern of the ATM gene caused by higher levels of this antisense oligonucleotide. And we could do, akin to our lysosomal functional assays in the Batten case, we could look at DNA damage functional assays in this case to show that our patients' cell fibroblasts, which showed a defect in their ability to mount a response to irradiation, could be rescued with treatment with one of our antisense oligonucleotide drugs. This is looking at levels of phospho-P53 and phospho-Cap1 as indicators of functional rescue. And with all of this, we were able to initiate investigational treatment for this child at age 2. And this trial now has been ongoing for the last 2.5 years. She had her 5th birthday recently. I'm pleased to say that she's tolerated the drug well. She appears to have minimal clinical symptoms so far, but this is actually not a trial that we're ready to read out yet. So I am telling you about it somewhat prematurely. It's too early to declare success because symptoms in this condition begin to manifest most strongly at around age 5. And so our motivation for entering here at age 2 was the chance to intervene pre-symptomatically. I think the readout for this trial will likely be in the next 12 to 18 months. So I look forward to reporting that out soon. In the meantime, though, I will note as an aside that 4 more children with this splice amenable ATM variant have surfaced. So unlike Milasen, which was a 1 in the world, we've found 4 other children globally that have the same variant. We have enrolled one of these children into a trial that's ongoing in collaboration with Boston and collaborators in Germany. And we're working hard to see how this might be made accessible to the other children as well. Our working with AT also connected us to the AT Children's Project, which allowed us to address a critical question in the field that we wanted to answer, which is to ask how many patients might be amenable to this strategy. One of the things that I'd like to impress on folks is that the strategy I've described sounds exciting, but it involves types of mutations that we have to define how common they are. Not every mutation, in fact, the great majority of mutations do not have the characteristics of these 2 mutations I've described, have these splice altering characteristics. So how can we systematically determine how many of these are really out there? And how do we actually set about finding them? Well, we worked with the AT Children's Project to answer this question. They had the foresight of setting up a registry to enroll 235 cases of AT. And they did whole genome sequencing on every single one of these individuals. Every single one had a clinically confirmed diagnosis of AT, and every single one of them underwent whole genome sequencing. And we worked through our logic to try and classify types of mutations with respect to their amenability to the strategy that we've developed. And I won't go into the details here other than to simplify and say that looking for mutations that cause gain of a splice site is most productive for predicting ASO amenability. There are some mutations that can lead to exon skipping that can sometimes be reversed with an ASO, so that puts it in a medium category. And then there are other mutations that are more challenging, but possible, and so we put those in the low category. And what did we find in this cohort of 235 cases? We found that 1, whole genome sequencing, which was performed in all these patients, allowed us to achieve a complete molecular diagnosis in 99% of them. And that is a significantly better yield than one gets using traditional clinical sequencing, which maxes out typically around 85 or 90%. What we found was that 36 of these individuals had at least one of their genetic variants potentially amenable to an ASO therapeutic approach. But perhaps soberingly, we found that they had a diversity of different mutations. So there was one recurrent variant that was found in 13 cases. So one ASO could be used to treat 13 of these children. The second most common mutation was found in 5, but treating all 36 cases, if one would want to be able to access all 36, would require up to 23 different antisense oligonucleotides. That's a lot of work. So I think this speaks to both the promise and the potential, but also some of the challenges in this space, that we're going to really have to lean heavily on finding ways and generating the data to show that this type of ASO can be used as a platform safely and reliably in order to really make this work the way we'd like it to. In thinking about the challenges to these really big questions, we've had the good fortune of partnering with many folks. We were new to the oligonucleotide therapy field when we met our patient in 2017. And we benefited greatly from interactions with the community, especially the Oligonucleotide Therapeutic Society, a public and private professional association with key leaders, including those shown here. Art Krieg and Anamika Arts-Marus are past presidents of the OTS. And one of the things that we did with the OTS is to put together a rare disease task force and draft a briefing document that would be meant to describe the opportunities that Milasen really represented, to highlight the capabilities and the limitations, the risks and the potential benefits of antisense oligonucleotides, and to summate this in a briefing document. And I encourage folks to visit this to visit the Oligonucleotide Therapeutics website if you'd like to read it. We've seen foundations now, like the remarkable n-Lorem Foundation, step in to try to help. n-Lorem is a nonprofit foundation founded by the founders of Ionis. Ionis is the company that very justifiably has the reputation for having done the most to advance the science of antisense oligonucleotides in the last several decades. And so it's fantastic. They have formed this foundation to charitably develop antisense oligonucleotides for ultra-rare conditions along the veins of the individualized application that I described for Milasen. And other academic centers, for instance, this one in Europe, the Dutch Center for RNA Medicine, have now launched with the specific aim of continuing to further work like this and to develop individualized medicines for patients there as well. And this one's headed up by Anamika, who I already mentioned in the previous slides, and Willeke van Moom. So this work also, to supplement these efforts, we ourselves have formed a coalition of academics, patients, government representatives as well, to help drive this conversation forward, to try to help coordinate conversations between these parties. This is the N of 1 Collaborative, a group that I'm really proud to be a founder of and to be associated with. And our goal within this group is to help catalyze conversations, to talk about standards for this field, and importantly, to share data. I think that it's critical that these are going to be disparate attempts from places all around the world. And there has to be really efficient ways for folks to communicate and to share learnings, both successes and missteps from these cases. So that's one of the primary purpose of this coalition. What's the landscape, how's the landscape reacting, the regulatory landscape reacting to this? Well, we've been really fortunate to have fantastic FDA regulatory engagement. In the last year, we've seen initial FDA guidances, brand new FDA guidances drafted specifically to cover this new precedent. And the FDA released 4 of these guidances: a procedural draft guidance, a toxicology standards draft guidance, a manufacturing guidance, and a clinical trial guidance, all on the topic of individualized antisense oligonucleotides in 2021. And they're committed to having continued conversations. These are just first drafts. There's certainly a lot of work to be done, but we find this really, honestly, we're really quite proud of the fact that this precedent seems to be standing up. And the FDA recognizes that this type of application is something that we'll need to have structures for, and they're committed to working with us to develop those structures. And these structures will be important not just for antisense oligonucleotides, but we can see the need for this in the tea leaves. Antisense oligonucleotides are the current most mature platform, but there are many other technologies that share some of these flexible characteristics: siRNAs, phosphomorpholinos, mRNAs, CRISPR genome editing. Many of these technologies act at the single mutation level and will require at some level similar individualized approaches. I'd like to close with one last case that allows me to highlight, I think, an important cautionary message. It's very apparent to us that this is an incredibly exciting area. We're incredibly invigorated by it. It's an incredibly appealing concept. But we also have to acknowledge that there are many hazards to navigate here, not just clinical and regulatory, but ethical. And much of our work focuses, for good reason, on individuals with orphan and very, very seriously debilitating or fatal conditions. But because of that, these are also populations that are quite vulnerable. And so we have to be careful not to raise expectations too confidently. And in each case, we take very careful adjudication of potential disease risks and treatment risks and try to weigh these together to come with what's an appropriate risk-benefit assessment. To make this real, I'd like to just close with this last clinical anecdote. In a project that I don't have time to go into great detail about, we launched an investigational antisense oligonucleotide for a very, very severe and life-threatening neonatal epilepsy associated with mutations in the KCNT1 channel. This is a mutation and a syndrome associated with infants having dozens of seizures per day. Half of these children die by the age of 3. And those that do survive past age 3 all suffer profound neurologic disability. And so this is really a terrible disease. And so we had selected this orphan condition as the focus of one of our early programs. And this slide here shows seizures in one of 2 patients that we treated with this condition. She was having between 5 and 15 seizures at the outset of the trial. Each of our doses are shown in the red stars, escalating doses, 10, 20, 30, 40 milligrams. And you can see is that the dose escalated through those first 50 days, that after dose 3 and dose 4, we saw something pretty shocking, a remarkable drop in seizure frequency, similar to, but arguably more dramatic than we'd seen in Milasen. And this persisted for some 50 days. And then gradually, as you see, rebounded and came back up to baseline over the ensuing 100 days. Now, why am I showing you this figure? Why are there no further red stars past day 50? The reason for that is that 15 days after day 50, we encountered a severe adverse event. We saw the development of ventricular enlargement and hydrocephalus. And this enlargement was significant, sufficiently significant that we decided for her protection to place a ventricular peritoneal shunt to relieve the buildup of pressure should her ventricles continue to expand. And thankfully, that monitoring and surgical procedure, which was all required an ICU stay and post-neurosurgical interventional care, that was not in the bargain for what our patient's family had signed up for. But thankfully, with that intervention, harm appeared to have been averted. But we've been very carefully monitoring the situation and not dosing further until we could assess what might be going on. And that's why we're seeing the slow return of seizures, because we've held further dosing since then. And I show this just to illustrate that there are now going to be uncertainties and unknown unknowns that are going to reveal themselves through these efforts. We actually know in as yet unpublished but publicly disclosed data from a Huntington's trial that antisense oligonucleotides developed for that trial also induced hydrocephalus and ventricular enlargement, and this time in adults. And so we're in careful discussions about mitigation strategies, changing the dose, considering alterations in the route of administration that might allow us to circumvent this, since we see there's both promise but also peril in these efforts. So with that, I'll stop. Thank the many stakeholders with whom the ideas I presented have been synthesized. Thank the many people involved in the work that I've tried to acknowledge along the way, especially acknowledging the support of Mila's Miracle Foundation, the AT Children's Project, and the Valeria Foundation, and several other philanthropies listed below. And I'll stop and welcome any questions or comments you may have.
Excellent. Thank you, Tim, for a wonderful talk. It's always great to hear your story and the message of both, I think, promise of what we might be able to do with some of these therapeutic technologies, and also the caution, I think, that comes along with exploring this area of drug development for these patients that, don't have any other options. We do have a couple of questions. So first, from David Dimmock, why do you think the oligo led to hydrocephalus? Do you think this is related to the infusion, drug toxicity, or just chance? Do you have any insight?
Hi, David. Thank you for that. That's a really important question. And the short and fairest answer is that we don't know yet, but it's obviously critically important to find out. And we have two agents, the Huntington's agent, arguably, and ours as well, which are going to be critical probes to really explore this and to figure out the parameters to avoid it in the future, right? But I can say more than that. I mean, that's a little too little. We do know, we looked for signs. Obviously there are things that are known to cause hydrocephalus, inflammatory insults, infections and so forth and so on. And there was no sign of infection. This happened actually in 2 out of 2 patients who received our investigational drug. It also happened in several patients. It was evident across the cohort in the Huntington's trial as well. We looked for signs of infection. There were no indications that infection had anything to do with it. There were no overt inflammatory changes. So normally inflammation, and obviously, as you know, in the central nervous system is typically indicated by high white counts and high protein levels and so forth. Our patients had really unremarkable white counts in their CSF. Their protein levels were pretty bland. In some cases, they trended up slightly, but really unremarkable by and large. Our best hunch at this point is that there is some low-grade aseptic meningitis, low-grade irritation, chemical inflammation, perhaps, that it seems to be associated with some antisense oligonucleotides. The generalizability of this is not yet clear. That is leading to defects in CSF reabsorption. But that's a model, it's not an answer, and there's a lot more work to be done.
Thank you, Tim. Since you suspect that it is unlikely that these compounds will be delivered by industry, do you anticipate that they will be funded, developed by foundations, academia, both, or other? I'm guessing likely this is going to be in the collaborative space, as it's typically had to be for rare disease.
Yeah, this is a critically important question. And I think that everyone's crystal ball on this is really, results hazy, unclear, right? But I would say certainly at the moment, I would characterize these as really hybrid research compassionate use efforts. But the research component can't be emphasized. This is highly experimental in nature. And so in that respect, I think that we should largely think of this as seeking funding and support from traditional research funding. NIH and NINDS are thinking very carefully about this. The challenge, though, it must be said, is that this is quite a challenge. This is an in-between space for NIH funding. Typically, the way that our system is set up, when something goes to trial, it's typically funded by venture capital, and it's funded by biotech and pharma investment. But this has a more researchy nature than much of that. So it's really sort of awkwardly in between. I would say, putting my futurist hat on, there are several different models that we've thought about and discussed with folks. One is that it's possible that this will be something that we'll never have a really clean model for, and that it'll always be supported by charity. And that's one possibility that is a little suboptimal in my mind. I hope we can come up with something more stable than that. Another possibility is that there is that this actually becomes not an effort that's this becomes an effort that's much more like a medical intervention, where we have insurance coverage to cover procedures and interventions like transplants and things of that nature. A medically intensive intervention for which there is some sort of Medicare coverage, Medicaid coverage for. Those are just a couple of the ideas that are out there. The last one that should be said is that there are some precedents for delivering platform-based individualized therapies like cancer vaccines or CAR T therapies, or even one would aspire to develop a series of COVID vaccines that vary from year to year to catch up with the latest variant. That there have been some situations where the FDA has seen fit to approve a process for making drugs that can be recycled and reused. So those are some of the ideas that are out there.
Yeah, I'm curious with that last statement, Tim. Do you envision that to be, disease specific then, or gene specific almost? So the platform like doing for ATM, or if there were other patent disease variants, right, where you think you could target more than one, changing the sequence, but keeping all else.
Yes, yeah,
the same.
Right. And I think that is one of the really appealing models, Margot. I think that if you think about what makes for a nice, coherent scientific story, one can imagine taking the 36 individuals that we identified in our AT cohort and developing the 23 ASOs necessary, and then combining them all in a platform trial where there's one disease, multiple patients, and multiple different interventions that are being assessed using the same safety and efficacy outcome measures. That is still, certainly by current drug trial standards, a very challenging trial, but it has sufficient scientific coherence on the efficacy endpoint with the same measures hopefully applicable to a large number of those individuals that you could imagine building an approval around that. But there are other even more creative ideas. Perhaps you could bucket trials that are designed all with a common seizure endpoint, even though there might be multiple genetic etiologies, et cetera, et cetera. There's lots of fascinating hypotheticals here. Each have their challenges, but we're just going to have to work it out.
Well, I know I'm really excited to see where we're able to go in this field and bring some help to these patients that really don't have any options. So I think we have to go. We're at the end of our hour here. So thank you everyone for coming, and thank you, Dr. Yu, for being here.
Thank you so much.